Hi, my name is Burcu Darst, and I'm an assistant professor in the Public Health Sciences Division at the Fred Hutch Cancer Center. I'm happy to give this talk today on population diversity in genetics and polygenic scores as part of the Introduction to Polygenic Scores course hosted by the Pan-African PGS Education and Research Initiative. First, I'd like to do a brief review of some of the concepts you learned in the first few lectures of this course to reinforce those lessons. Polygenic scores quantify a genetic predisposition to a heritable trait, and these are calculated as a sum of genetic alleles weighted by corresponding variant-specific effect sizes. Some of the terminologies that we often use include polygenic risk scores, or PRS, and genetic risk scores, or GRS. These in particular are referring to the genetic contribution to disease risk. The term polygenic scores, or PGS, is an umbrella term that can include PRS or GRS, but can also be referring to the genetic contribution of any phenotype, such as a biomarker or height, where the term risk may not necessarily be appropriate. There are a few potential clinical utilities for PRS that have been described. This includes using PRS to inform disease screening, including whether or not to initiate disease screening, how frequently it can be done, and how to interpret results. PRS could also inform therapeutic interventions, and they could inform life planning in the event that there is not a clinical intervention available for a given disease at the time. You'll note on all of my slides that whenever I include references, I try to include the full reference. That way, if we don't have time to really go into a topic in too much depth, you can look at the reference and learn more about that topic on your own. Polygenic risk scores are calculated as a weighted sum of genetic effect alleles weighted by corresponding variant-specific effect sizes. So in this equation here, G is the number of effect alleles for a variant M and individual I, and beta is the weight of variant M. This is summed over all the variants that we look at, so each individual ends up getting a polygenic risk score. An example of how this can be calculated is shown here. So in this figure, we have the results from a genome-wide association study, or a GWAS study. We take all of the significant variants from it. You can see here there are 4 variants that have p-values that are below the significance threshold. We extract from those the effect alleles, the non-effect alleles, the beta, the effect for the specific allele. And you can see by looking at the p-value that they all meet the significance threshold. We then, in an external data sample, can calculate the polygenic scores for these individuals based on the information that we see in the discovery GWAS. So here we're showing for these 2 individuals the number of effect alleles that they carry for these 4 SNPs that are being considered in this table here. You can see for person 1, they carry 1 copy of the effect allele for SNP 1. They carry 1 copy of the effect allele for SNP 2, 0 copies of the effect allele for SNP 3, and 2 copies for the effect allele of SNP 4. We can plug into this equation the weights for each of these SNPs, and then we can calculate their polygenic risk score, and you see on the right side of this equation what the polygenic risk score is for each of the individuals that we're looking at. Once we have these polygenic risk scores calculated for all of the individuals in our data set, we can plot the distribution of the PRS in cases, as shown by the solid line here, and in controls, as shown by the dotted line here. And you can see that our PRS is doing well because the distribution of the PRS is a little bit higher in cases than it is in controls. There's a couple ways that we like to analyze polygenic risk scores. We can analyze them as a continuous predictor, where we'd often standardize the PRS, and then we can interpret the results such as each standard deviation unit increase in PRS is associated with X-fold increased disease risk. Another way that we like to evaluate polygenic risk scores is as a categorical predictor. So the way that we do this is to stratify all of our participants into PRS categories. So here I'm showing that we stratified our participants into PRS deciles. Those decile cutoffs are determined by the PRS distribution in controls. We can then compare the number of cases in controls in the top PRS decile versus the median 40 to 60% category and calculate odds of disease. We would then interpret it as individuals in the highest 90 to 100% PRS category have X-fold higher odds of disease than those in the median 40 to 60% PRS category. There's a few different ways that we like to evaluate the performance of polygenic risk scores. If we're looking at a dichotomous outcome, we can use logistic regression, and the metrics that we would be looking for are odds ratios and p-values for the effect of the PRS on the trait. We can also evaluate the area under the curve, or AUC. We like to look at the net reclassification index, or NRI, and you can also look at variance explained, or the pseudo R-square. If you have a continuous outcome, then you can use linear regression, and again, you would look at the effect on the trait, looking at the beta and p-value, and then you can look at variance explained, or the R-square. In all of these analyses, we adjust for age, sex, and population stratification measured by principal components of ancestry. There are a few different factors that impact the maximum achievable PGS performance. So in this figure here, it's showing the sample size of discovery GWAS used to develop a polygenic score across 3 different samples, 3 different cancers: colorectal, breast, and prostate cancer. On the x-axis is the different sample sizes of these discovery GWAS, and on the y-axis is the AUC. The red dotted line across each of these figures is the maximum achievable AUC for any given polygenic score. You can see that the maximum achievable AUC is highest for prostate cancer, and the heritability, as I've annotated on this figure, is highest for prostate cancer compared to any of these other cancers that we're looking at. You'll also note that the AUC that you can achieve is higher with a larger GWAS discovery sample size. So having a large sample size is really important for being able to develop a very accurate polygenic score. And having high heritability is also important for being able to develop a polygenic score that's really effective. That is similarly shown in this figure shown here, where instead of looking at AUC, we're looking at relative risks of disease. And you can see, similar to before, that the higher the sample size and the higher the heritability, the higher the AUC you can achieve for a polygenic score. Another thing that I want to point out in this figure is that we have these heritabilities annotated here, but I just want to note that these heritabilities are from a twin study in Nordic countries, and heritability is a population parameter, so it depends on population-specific factors such as allele frequencies, variant effects, and environmental factors, which can differ between populations. So these heritabilities do give us a good sense of how heritable these different cancers may be, but it's important to note that the exact heritability of these given cancers may vary between different populations. If you're interested to learn more about the development and evaluation of polygenic scores, take a look at the references that I've cited here. There's these 2 different papers. Both of them have figures that are pretty similar to this one shown here, and they give you really nice walkthroughs of how to develop polygenic risk scores, how they're evaluated, different criteria that are used. So take a look at these if you're interested to learn more about this topic. There's been a lack of diversity in GWAS studies over the years that could contribute to health disparities. This figure here is showing that over the years, the overwhelming majority of participants who have been included in GWAS studies have been from European ancestry populations. However, that is not at all reflective of the global population, where we see that only about 15% of the global population is from European ancestry populations. Consequently, when we develop polygenic models based on GWAS data, we see that the predictive accuracy is increasingly poor in populations who are more ancestrally distant from European ancestry, and we often see the lowest predictive ability in African ancestry populations. This could potentially exacerbate existing health disparities, where we see, for instance, in the U.S., Black or African American individuals have the highest incidence and mortality rates for prostate cancer compared to any other population seen here. The way we have determined that PGS performance varies between populations is by grouping individuals into broad populations, typically defined by principal component analysis as shown in Panel A, and evaluating PGS accuracy at the population level as shown in Panel B. However, this has several limitations. It ignores inter-individual variability within groups. Group boundaries depend on algorithms and reference panels, which can lead to arbitrarily defined boundaries between populations. And many individuals may not be assigned a group if certain populations are not represented in the reference panel. This paper by Ding et al. Suggests that evaluating PGS accuracy at the individual level circumvents these issues and provides more information on PGS accuracy, allowing you to see that PGS accuracy can also vary within populations due to the genetic variation that exists within populations. Panel C demonstrates that instead of assigning individuals into groups, they can be assigned a value indicating their genetic distance from the center of the training population used to develop the PGS. As shown in Panel D, you can then use this information to see how PGS accuracy changes by genetic distance from the training population. This paper went on to demonstrate how individual-level PGS accuracy can be evaluated in the UCLA Atlas Biobank using a PGS of height as an example. Here they show that within each of these genetically inferred ancestry groups, represented by the different colors in this plot, PGS accuracy varies widely, with accuracy decreasing with increased genetic distance from the training population. Although this approach is much more informative than the typical population-level assessment of PGS accuracy, it is important to note that this is often not possible to implement since most PGS are trained on GWAS summary statistics across many studies where individual-level data are not available to be able to calculate the genetic distance to the training population. There are several reasons why PGS accuracy varies by genetic ancestry and by genetic distance to the training population. As represented in Panel C on the left, the effect of the causal allele, represented by the diamond, may vary between the discovery and target population. In this case, using variant effects determined in the discovery population could decrease PGS accuracy in the target population. Another reason PGS accuracy varies between populations is that the linkage disequilibrium may vary between populations. In the top of Panel D, the most significant variant identified, represented by the yellow circle, is strongly correlated with the causal variant, represented by the red diamond, in the discovery sample. However, the bottom of Panel D shows that this variant is weakly associated with the causal variant, represented by the green diamond, in the target sample. In this case, the PGS developed from the discovery sample is not capturing the causal variant in the target sample, which could decrease PGS performance. Another reason why PGS accuracy can vary by genetic ancestry is that genetic ancestry can be correlated with non-genetic risk factors, such as environmental factors or social determinants of health. Even if genetic ancestry is accounted for in analyses, residual bias could impact PGS accuracy across populations. These non-genetic factors can also serve as mediators of the effect of PGS on disease risk, with PGS having differential performance in individuals with certain exposures. As such, it's important to consider PGS in the greater context of other risk factors and potential mediators for a given trait. Including individuals across diverse genetic ancestries can be a powerful way to improve the performance of PGS across populations. For instance, GWAS results across multiple populations can be leveraged to narrow the genetic regions that is likely to contain the causal variant using an approach called statistical fine mapping. In this top figure here, you can see that in population 1, the most significant variant in this region, represented by the yellow diamond, is different than the most significant variant identified in population 2, shown in the second figure. In population 1, these two variants are weakly correlated, as indicated by the green dot on the left in the first figure, which suggests low LD. However, in population 2, these two variants are strongly correlated, as indicated by the rightmost red dot in the second figure, which suggests strong LD. We can leverage these results and these population structures to narrow down the region that is most likely to contain the causal variant, which likely lies between these two variants. Including causal variants in PGS is a notable way that PGS accuracy could be improved, and this can be facilitated by including individuals from diverse genetic ancestries in discovery GWAS used to develop PGS. In recent years, many PGS approaches have emerged that leverage diverse genetic ancestries to improve PGS. This table, which was abbreviated by a review from Kachuri et al., summarizes some of these methods, most of which take as input GWAS summary statistics and LD matrices. Be sure to check out the review and the corresponding references to any of these approaches you may be interested in to learn more about these methods. So if you have a research question and you'd like to develop a PGS in your data, how do you go about identifying a PGS? One amazing resource is the PGS catalog, which is a fairly comprehensive catalog of published PGS to date. Using an existing PGS based on a large sample size can be a simple and powerful approach to conducting PGS investigations. The PGS catalog has nearly 5,000 PGS covering over 6,000 traits, and for each PGS it provides information about the study population used to develop the PGS and the study populations that the PGS has been evaluated in, as well as performance metrics. It's really a great way to identify a PGS that may be an ideal fit for your study. However, the PGS catalog was recently used to demonstrate that about 88% of PGS have been developed in populations of European ancestries, even for traits that disproportionately affect populations of non-European ancestries, such as type 2 diabetes, as shown in this inset figure. Further, only 87 PGS of the about 3,500 PGS in the catalog at the time of the study have been developed in African populations, suggesting the lack of global representation in PGS development. This demonstrates the crucial need to include more diverse participants in PGS development to ensure that PGS findings will be impactful and effective for everyone. Next, I'm going to give a few applied examples of PRS projects we've conducted in prostate cancer. Given the huge health disparity that prostate cancer represents, one of the main goals of our work is to understand prostate cancer risk in African ancestry populations and identify genetic factors that could mitigate risk in these populations. So the first example that I'll give is a multi-ancestry GWAS meta-analysis of prostate cancer that we conducted a few years ago. This analysis included over 200,000 men across the 4 populations shown here, and these are broadly defined populations. We conducted our analyses separately in each of these populations first, and then we meta-analyzed results across the populations to get multi-ancestry results. We performed statistical find mapping in each of the populations to identify regions that are more likely to be causally associated with prostate cancer risk, and then we combined in fixed effects meta-analyses the GWAS results from each of these studies to obtain multi-ancestry conditional summary statistics. We found 86 novel risk variants that were associated with prostate cancer risk. For 62 known risk regions, we were able to identify stronger markers of risk using the find mapping approach that I mentioned, and 121 other known variants remained the lead signal in their regions, which brought the total to 269 prostate cancer risk variants that we knew of at the time that we conducted this study. In total, these 269 variants accounted for about 34 to about 43% of the familial relative risk of prostate cancer, meaning that we're able to account for a lot of the genetic variation of prostate cancer, but there's still more that remains to be accounted for. We used these 269 variants to calculate a polygenic risk score for prostate cancer. This is done in about 500,000 individuals who were independent from our discovery GWAS sample. And these 500,000 individuals were across 6 different large biobanks and across 3 different populations as shown in this table. And here I'm first showing the results separately for each of the biobanks and each of the populations so that you can see that the performance of the PRS, just looking at these different odds ratios, was actually fairly comparable across different studies and different populations. I'm also highlighting here that we did have a couple studies that included participants from Africa. So if you look at the purple and blue in these figures, you can see that the odds ratios are pretty good in those studies too. So we do see pretty good performance of our prostate cancer PRS in participants who are from Africa. When we meta-analyze results across these different studies, you can see that the odds of prostate cancer associated with the PRS range from about 2.8 in African ancestry individuals to up to 3.8 in European ancestry individuals. And that's looking at the odds of prostate cancer in men in the top PRS decile, the 90 to 100% category, relative to men in the 40 to 60% PRS category. So this is suggesting that although it is doing fairly well across all the populations we're looking at, we do see lower performance in the African ancestry populations, suggesting that we still have room for improvement for our polygenic risk score. Just to give you a sense of what polygenic risk score performance looks like across populations for other traits, here I'm showing this example of a polygenic risk score for breast cancer. And in this example, the PRS that's being evaluated was developed in a European ancestry sample. That PRS was then tested across these 4 different populations shown here. And here this figure shows the results across those 4 different populations. You can see the performance was highest in European ancestry individuals. It was relatively decent in Asian and Latino individuals, and lowest in African ancestry individuals. This is not unexpected since the discovery population was limited to European ancestry individuals, so it's not a very representative sample of the individuals being tested in the test data set. I'll also note that the odds ratios seen here go up to about 2.5 at their best, which is a bit lower than what we're seeing in prostate cancer. Another example is a polygenic risk score for type 2 diabetes. This polygenic risk score was developed in this multi-ancestry sample shown here in the top table across these 4 different populations. The PRS developed from that study was then tested across these 5 populations shown in the bottom table, and results for that are represented in this figure. So here you can see that the PRS does pretty well in both European and Asian ancestry individuals. And again, we see the lowest performance in African ancestry individuals. Although there are African ancestry individuals included in the discovery data set, the sample size is not as big as what we're seeing for Asian and European ancestry individuals, and could be the reason why the performance is lower in African ancestry populations. So this is really just reiterating how important it is to ensure that there's a really good representation of all the populations that you're interested in the discovery data set. Next, I'm going to talk about some ancestry-specific variants that we've included in our prostate cancer PRS. I'm just going to talk about this one example, and this is an African ancestry-specific variant that is in the 8q24 region that I'll refer to as rs727 for short. So this variant was initially identified in this GWAS of African ancestry participants on prostate cancer risk a few years ago. This included 10,000 cases and 10,000 controls. The strongest by far variant that was identified is this signal shown here, this rs727 variant. This is a fairly common variant. It's carried by about 11% of African ancestry men. And the odds ratio is 2.3, which for a GWAS study is really large. Usually in GWAS studies, the variants that we identify have odds ratios of maybe 1.1 or 1.2, so this is a really large odds ratio. This particular variant is in a non-coding intergenic region near PCAT1 and prostate cancer-associated long non-coding RNAs. It lies in a prostate cancer-specific enhancer, which interacts with long non-coding RNA promoters. So even though it is in a non-coding region, it could have some potential biological function that is relevant to prostate cancer risk. So we wanted to further understand this particular variant and its association with prostate cancer risk. So we did this analysis with just this one variant. This is not polygenic risk score results that I'm showing right now. And these results are just showing that with a stronger family history of prostate cancer, there was a larger odds ratio associated with prostate cancer for this one variant. We also see that this variant is increasingly common and associated with a larger risk of more aggressive disease. So the more aggressive the disease, the more common the variant is, and the larger the odds ratio we see. We then looked at the absolute risk of prostate cancer by age, carrier status for this variant, and family history. So here on the y-axis we have absolute risks. On the x-axis we have age. The different colored lines indicate different carrier statuses for this variant. And then the solid line is showing absolute risks for family history negative men, and the dotted line is absolute risks in family history positive men. So just focusing first on absolute risk in family history positive men, you can see that the absolute risk of prostate cancer is about 9% in family history positive men who have no copies of the allele by age 60. That goes up to 21% in family history positive men who carry one copy of the allele, and 38% in family history positive men who carry two copies of the allele. The highest absolute risk we observe is 80% absolute risk of prostate cancer by age 85 in family history positive men who carry two copies of the risk allele. And I also just want to point out that the absolute risk of prostate cancer is about the same in family history negative men who carry one copy of the allele as it is family history positive men who carry no copies of the allele, which is really just suggesting that this variant alone conveys about the same amount of risk as having family history of prostate cancer, and that looking at these two pieces of information together, family history status and carrier status for this rs727 variant, is really equally important to do. We also wanted to investigate the contribution of this variant to prostate cancer risk relative to all other known prostate cancer risk variants. At the time of this study, there were 180 total known prostate cancer risk variants, which collectively accounted for about 28% of the familial relative risk in African ancestry men. However, this variant alone accounts for about 9% of the familial relative risk, which is about a third of the familial relative risk accounted for by all known prostate cancer risk variants. Another way we addressed this question was by developing a polygenic risk score, including and excluding our 8q24 variant, looking first at the full GRS with all 180 variants represented by the gray lines in this bar. The odds of prostate cancer for those in the top PRS decile is about 2.75. When we exclude our rs727 variant, that drops down to 2.25, clearly displaying that a large proportion of the effect captured by the full PRS can be attributed just to this rs727 variant alone. We also found that the risk allele frequency of rs727 was highly variable between different African ancestry populations, with the lowest risk allele frequency being about 2.5% in Akoli men from Uganda, and the highest being about 15% in men from the Democratic Republic of the Congo. Looking at African ancestry men in North America, we saw less variability, with the risk allele frequency ranging from about 4 to 8%. This figure shows that the risk allele frequency of rs727 is much higher in men with more global African ancestry, which we determine using genetic information, as shown in the right side of the figure, and less common in those with less African ancestry, shown on the left side of the figure. This really highlights that this is truly an African ancestry specific variant, and given that we know it's a really important risk factor for prostate cancer, it's one that we wouldn't have been able to identify if we weren't able to include African ancestry men into our prostate cancer GWAS analysis. The last topic that I want to touch on is incorporating polygenic risk scores with rare genetic variants. So here we're asking the question, could rare variants improve the discriminability of polygenic risk scores? We know that pathogenic variants are associated with substantial disease risk for some disease, including prostate cancer. Pathogenic variants are typically defined as rare variants with a minor allele frequency less than about 1% that have some deleterious functional consequence. This could include protein truncating variants or splice altering variants, for example. However, it's really challenging to study rare variants because they're so rare. Individual variants alone are really unlikely to substantially be able to contribute to population-level risk prediction. So instead, what we often do are to conduct these gene burden tests, where instead of testing one variant at a time, we'll aggregate variants across a gene or some specified genetic region, and then see if carrier status for variants for any of these rare pathogenic variants in a given gene is associated with, for instance, prostate cancer risk. There are a couple really nice examples in breast cancer where the effect of PRS on breast cancer has been shown to be augmented by carrier status for rare pathogenic variants. So looking, for instance, in this figure on the left, you can see that the breast cancer risk by age 80 among CHEK2 carriers, shown by the dark blue curve there, Absolute risk can range from about 5 or so percent in check 2 carriers who have a low PRS, all the way up to about 60% in check 2 carriers who have high PRS. And this is really important, especially for breast cancer, because in the US, guidelines recommend that women who have greater than 20% lifetime risk of breast cancer get annual breast MRIs. So this has implications for screening practices. And for check 2 carriers, it's really unclear whether or not they should be getting these annual breast MRIs, because their risk kind of straddles. So it's right around this 20%. But if you're also to take into consideration their polygenic risk score, it can help you make a decision about what someone's actual risk of breast cancer is. So we know that if they're a check 2 carrier and they have high PRS, they might be a good candidate for receiving annual breast MRIs. So we wanted to look at this in prostate cancer too, to see if carrier status for rare variants and information about polygenic risk score in aggregate could improve prediction of prostate cancer risk. So we first conducted this study in the UK Biobank, and this was across a large number of European ancestry men. Here we looked at carrier status for rare pathogenic, likely pathogenic, or deleterious variants across these 4 genes: HUXB13, BRCA2, ATM, and CHEK2, and these are known prostate cancer risk genes, which is why we decided to look at these in particular. So in this figure, on the x-axis, we're showing the PRS categories. On the y-axis, we're showing odds of prostate cancer. In the light blue, we're showing odds of prostate cancer in carriers. And in the dark blue, we're showing odds of prostate cancer in non-carriers. So you can see, for instance, that the odds of prostate cancer in non-carriers who have a high PRS, it's about the same as what we see in carriers who have median PRS. It's right about 3 or so, threefold higher than what we see in non-carriers who have median PRS. We also found the absolute risk of prostate cancer by age 85 in carriers was about 26%, and in non-carriers it was about 11%. But again, if you incorporate polygenic risk score information into those absolute risk estimates, you can actually see that among non-carriers, absolute risks range from about maybe 2% all the way up to about 30%, just looking at the solid lines in this figure here. But in carriers, they range from maybe about 10% all the way up to about 55%, looking at the dotted lines in this figure. So here you can see that the absolute risk of prostate cancer depends not only on carrier status, but polygenic risk score information, and that there can actually be a pretty wide range in absolute risks that depend on having both of these genetic risk factors, information about both genetic risk factors. Subsequently, we conducted a similar study in a large sample of African ancestry men. This is done in over 3,000 men. Most of them were African American men, but it also included a large number of men from Uganda. And here we looked at carrier status for rare pathogenic variants in BRCA2, ATM, NBN, and POLB2, as those have been shown to be big risk factors for prostate cancer in African ancestry populations. And we saw similar results in this study. So now here on the x-axis, again, we have the PRS categories, but it's split by tertiles because our sample size was a little bit smaller than what we had in the previous UK Biobank analysis. Odds ratio is still on the y-axis here. And now in green, we're showing odds of prostate cancer among carriers, and in black, we're showing odds of prostate cancer in non-carriers. And you can see that the odds of prostate cancer in non-carriers who have high PRS is about the same as what we observe in carriers who have low PRS. So again, really just conveying that having both pieces of information is really informative, that you can be a carrier and still have relatively low risk of prostate cancer if you have a low PRS. Similar to the other study, again, we looked at absolute risk of prostate cancer by age 85, and we found that it was about 48% in this population in carriers, and in non-carriers it was about 15% by age 85. But again, if you look at those absolute risks among carriers, when you incorporate information about polygenic risk scores, you can actually see that there's a wide range of risks that depend on what their PRS is. So in non-carriers, shown by the dotted lines in this absolute risk figure, you can see that the absolute risk range from about 5% all the way up to about 25% in non-carriers, depending on what their PRS is. And in carriers, shown by the solid lines here, their absolute risks range from about 15% in men with low PRS all the way up to about 70% in men who are carriers and have high PRS. So across these studies, we're really seeing that considering rare variant carrier status and PRS in conjunction is likely to improve risk stratification. In summary, I hope one of the main takeaways from this presentation today is that it is crucial to include diverse populations in genetic investigations. As we showed, this can improve PGS models across populations and uncover novel biology that we would not otherwise be able to identify. This can also enable the identification of population-specific variants, which can be really important for polygenic risk score prediction. There's strong potential for genetics to reduce health disparities via screening and disease management. We found that a multi-ancestry PRS of prostate cancer was a valid predictor. It was highly replicated across diverse populations. Population-specific genetic variants can also improve PGS accuracy and be really important for disease screening and disease management. One example that we saw of this was the AQ24 African ancestry specific variant that was really important for the polygenic risk score for prostate cancer. But more research is needed to improve the predictive ability of PGS across populations. In most examples of PRS that we look at, we often see that there's lowest predictive ability in African ancestry populations, and that really demonstrates the need to improve and increase the sample sizes of representation of African ancestry populations in our discovery GWAS used to develop these PRS. There are many limitations for PRS right now in terms of clinical utility. Clinical utility really needs to be thoroughly evaluated before we implement PGS in a clinical setting. This is a very costly thing to do, and it can be a really long-term study, so it's a challenging thing to do. But there are several studies right now that are working on doing these things and evaluating PGS in a prospective clinical setting. There also needs to be more physician and public education regarding PGS so that we can ensure that it's appropriately used in a clinical setting and understood by the physician and the patient. And then, of course, there's this issue of between population transferability, which really needs to be improved so that we do not increase the risk of health disparities across populations. The research that I presented today was really on the behalf of many, many people and funding sources who have contributed to this work over the years. So I'd like to thank all of the people who contributed to this. And thank you so much for your attention today and for the opportunity to present this to all of you. I really look forward to our Q&A session and getting to interact with you all.